Hi dear children, today I will be taking the 19th chapter, Excretory Products and their Excretion, and the topics I am going to handle in this video: Excretory Products, Human Excretory System and Nephron. So let's get started. So now in this slide we are going to classify the different animals into three categories based on their excretory products. They are classified as ammonotelic, ureotelic and uricotelic. So once again, animals are classified based on the major nitrogenous excretory product into ammonotelic, ureotelic and uricotelic. So the major nitrogenous waste excreted in ammonotelic animals is ammonia. In ureotelic it is urea, and in uricotelic it is uric acid. So the first difference we get with respect to the major nitrogenous waste excreted by these three categories of animals. In ammonotelic it is ammonia, in ureotelic it is urea, and in uricotelic the major nitrogenous waste is uric acid. Of course, salt and water is also other constituents of the excretory products. Now talking about the toxicity with this major nitrogenous wastes, ammonia is very toxic. Urea is less toxic, whereas uric acid is the least toxic. Now the water for disposal. For disposing ammonia we need a lot of water, requires a lot of water. And definitely we can correlate with their habitat. They should reside in water because they need a lot of water to dispose this ammonia. It's very toxic, cannot be retained in body. So as and when this ammonia is found, it has to be disposed of. So they should have access to water. So definitely they will be living in the water environment. Then this is recovering. The second category, ureotelic, requires less amount of water, and this requires the least or minimal amount of water. We have no access also to water. Then the fourth point or parameter is the role of the kidneys. In the ammonotelic animals, the kidneys, there is no role for the kidneys. No role for kidneys. The kidneys have no role, because as and when the ammonium ions are formed, they are diffused out through the body surface or through the gills. So as and when the ammonium ions are formed, they are diffused out from the body, and there is no role of the kidneys. Whereas in ureotelic and uricotelic, kidneys play a very important role both for excretion and osmoregulation. We will be dealing that later. What is osmoregulation? As of now, let us concentrate on the criteria of classification, and lastly I will put down the examples. The examples come under ammonotelic are definitely those organisms that reside in water. They are the aquatic insects, amphibians, and most of the fishes, especially the bony fishes. Example for ureotelic is us, man, and all mammals. We all are ureotelic. The major constituent is urea. And uricotelic are the insects in the air, the reptiles and birds. It's easy for you to identify these because the excretory product is in the form of a white paste or white pellet. Have you seen the excreta of lizards? In addition to the undigested waste that comes out, a white paste. Have you seen the excreta of birds? You can see along with the black content of the undigested food that is coming out, there is a white paste. and insects white crystals or pellets, not crystals, some pellets. So, they do not have access to water and they cannot afford to lose water by way of excretion, and therefore, they excrete it in the form of pellets or paste, and usually it is white in color, and not only that, it is very least toxic. So, it is not frequent, they do not excrete frequently. When they excrete along with the undigested waste that comes out through the anus, that excretion via the kidneys come out in the form of paste or pellets. So, once again reiterating the differences between the three categories of animals based on the excretory product. First difference is based on the major nitrogenous waste excreted. In the first category it is ammonia, in ureotelic it is urea, and uricotelic it is uric acid, and hence the name we get. Then the second parameter is toxicity. This waste product ammonia is very toxic, whereas urea is less toxic and uric acid is the least toxic. Third, the water for disposal. For ammonia it requires a lot of water, whereas for urea less water is required, whereas for uricotelic least or minimal water is required for its disposal. Fourth is the role of the kidneys. Here in ammonotelic, kidneys do not play any role in excretion. Only as and when the ammonia is formed, it is diffused out through the body surface or through the gills. And in ureotelic and uricotelic, kidneys play a very important role in two processes, excretion as well as osmoregulation. And finally, winding up with the examples for ammonotelic is aquatic insects, aquatic amphibians and fishes, mainly the bony fishes. Then ureotelic is the examples are us, mammals, and for uricotelic, insects, reptiles and birds. You can remember with respect to the pellets or the paste, white paste, birds, lizards, so the reptiles and insects. Moving on to the next slide, I hope you have understood this template. Next, I am going to deal with the human excretory system. We have learnt in lower class, it comprises a pair of kidney, a pair of ureters, a single urinary bladder, and a urethra. The kidneys, as you know, it is bean-shaped structure, and where it is located in the lower abdomen towards the dorsal side, not on the ventral side, towards the dorsal side. And a very important gland is located above it, that is the adrenal glands. So, it becomes part and parcel of the kidney. When you are drawing kidney, definitely there will be a cup like structure over it, the adrenal gland. This kidney, there are two of them, and arising from the concavity of the kidney are two tubes. These two tubes are called the ureter, and the ureter opens, the right and left opens into the sac called the urinary bladder, and the urinary bladder opens out through an opening or through a tube called urethra. The kidney is covered by a tough capsule. Very tough capsule covers the kidney, and not only that, we can divide it into two regions, outer cortex and inner medulla. So, I am doing the bifurcation in this chart itself, in this diagram. You can see there is the outer cortex and the inner medulla. So, this region is a cortex and the inner region is called the medulla. So, once again a briefing of the human excretory system. It comprises a pair of kidney, and from each kidney arise a pair of ureter, and these two ureters opens into a sac called urinary bladder, and the urinary bladder opens out through the urethra. And the location of the kidney is located in the lower abdomen on the dorsal side. Kidney is covered by a tough capsule and it is divided into two regions, outer cortex and inner medulla. Now, moving into the details of the kidney. If this is the kidney, it's bean-shaped structure. I told you it's covered by a tough capsule, and you can demarcate two regions, cortex and medulla. So here I'll demarcate it. The outer cortex and the inner medulla. Now in the concavity of each kidney there is a notch. So let this be a notch, an opening. This is called the hilum. And inner to the hilum you can see a wide or broad funnel like structure. I am just drawing like a funnel. See, now you got a funnel. Let me take another color to demarcate the funnel. A funnel like structure. This broad funnel like structure is called the renal pelvis. And look at the margin I have drawn. It is smooth margin. No, it is not smooth. It is thrown into projections. There is a name for that, projections. So I will just redraw the diagram. So I am going to draw the funnel with red color. And I told you how is the margin. It is not smooth as I showed you earlier. It is thrown into projections. See, one projection, second projection, third projection, fourth projection, fifth projection, sixth projection. See this funnel. Okay. So what I have demarcated is the renal pelvis. Yeah? And these projections, see what I have drawn like M-shaped structure. These projections are called calyx. In plural, calyces. Okay. Into these calyx, there are cone-shaped masses projecting in. So cone-shaped masses I will draw with blue color. So cone-shaped, like pyramids, they are projecting into these calyces. One pyramid, this is the second pyramid, third pyramid or cone, fourth, fifth, and so on. Okay. So into these calyces, what is projecting? Cone-shaped masses, and these cone-shaped masses which are like pyramids are called the medullary pyramids. So from this what we can guess the parts of the medulla. So the parts of the medulla, of course, it includes the renal pelvis, then the medullary pyramids, calyces. So all in blue is indicative of the medullary region. Okay, all that is blue color is the medullary region. Okay, so the medulla comprises the renal pelvis and the medullary pyramids. And where is the medullary pyramid embedded or projecting into? It is projecting into the calyx. Okay, see the calyx, I am just showing the calyx projections. Calyx. So only in yellow I am indicating the calyx, the projections. The margin of the renal pelvis is not smooth, it has thrown into projections called calyx. So the calyx I have highlighted in yellow color. See the projection into that the the medullary pyramids are projecting. Now talking about the cortex. The remaining portion is the cortex. So that part I am going to highlight with a highlighter. So what I am shading is the cortex. See, the remaining part is the cortex. See, I am drawing the cortex with patience because one region I am going to define. So cortex, I am shading the cortex. So I have covered the cortex, but it is not complete. This intervening, you can see intervening regions between the medullary pyramids. See, and if I highlight it, so this region, so this gap, this gap, this gap, this gap. This intervening space or regions in between the medullary pyramid, what are they? Are they medulla or cortex? They are part of the cortex. So the cortex is not just a C-shaped structure at the periphery, it extends in between the medullary pyramid. So I am going to extend it. With the highlighter, I am going to extend it. And I am going to give a darker shade of green. So now what I have shaded is only the cortex, and it is not just a C-shaped structure on the periphery, it extends in between the medullary pyramid. See, between these two medullary pyramids I extended. Again between these two medullary pyramid I extended the shading. Again between the two medullary pyramid I have extended. Again between these two medullary pyramid I have extended. Again here I have extended. So here also I am extending. So this region, to define it, the region between two medullary pyramids. So how many medullary pyramids are there? 1, 2, 3, 4, 5, 6. In my diagram I have given 6 medullary pyramids, and you can see the region between the two medullary pyramids, 1 and 2, there is a region gap, 2 and 3 there is a gap, 3 and 4 there is a gap, or 4 and 5 there is a gap, and 5 and 6 there is a gap, that the cortex extends. See, the cortex extends into between the two medullary pyramid, and that region is known as column of Bertini. Very, very important. Column of Bertini. So what is column of Bertini? The cortex extends in between adjacent medullary pyramids. That region is known as column of Bertini. So with that I have completed the structure of a kidney. So once again, kidney is bean shaped, surrounded by a capsule which is very tough. There are two regions, outer cortex and inner medulla. Then talking about the notch, at the concavity there is a notch, there is an opening, and that opening is called the hilum. Into the hilum what and all enters? The ureter, the blood vessel, the nerves enters. So 3 tubes, okay, they enters through the hilum. Now this hilum opens into a wide or broad funnel like structure, and that funnel like structure is known as the renal pelvis. And the funnel, you can see how is the margin of the funnel, it is thrown into projections. And what are those projections called? Calyx. What are those projections called? It is called calyx, and I have highlighted in yellow color. Into these calyxes there are masses, pyramid masses called medullary pyramids that are projecting. See, I am highlighting, these are medullary pyramids. So the medulla is renal pelvis along with the medullary pyramids. So the remaining part is the cortex. So the outer part is the cortex, and the region between the two medullary pyramids or between adjacent medullary pyramid, that region of the cortex is known as column of Bertini. I hope you have understood the structure of the kidney, that is through the longitudinal section. Next, each kidney is comprised of many millions of structural units called nephron. In nephron also you have studied the structure in lower classes. Now let us deal with the structure in detail. The nephron basically comprises two parts. The two parts are the glomerulus and the renal tubule. Now I am going to draw the renal tubule. It starts with the Bowman's capsule. And ends at the collecting duct. Very important, the renal tubule, it starts with the Bowman's capsule, which is indicated here, and ends with the collecting duct. So, I am going to draw. The Bowman's capsule is a double-walled cup. Okay, and then it leads to a highly convoluted structure, and this convoluted structure or coil structure is known as the PCT, Proximal Convoluted Tubule. This is the Bowman's capsule. Then this PCT leads to a hairpin loop. You know the pins used to tie the hair up, hairpin loop, like a loop which resembles a hairpin. This is known as Henley's loop. And Henley's loop has both two limbs, the descending limb and the ascending limb. Then again another convoluted structure, highly convoluted structure, and this is at the distal end, so this is called DCT, distal convoluted tubule, and finally it opens into a straight tube called the collecting duct, which receives many nephrons. Clear? Once again, I have indicated the renal tubule in blue color. It starts with the Bowman's capsule and ends at the collecting duct. So, I drew the Bowman's capsule once again with another color over it, a darker shade of blue. It starts with the Bowman's capsule, it's a double wall cup. Okay, then it goes to a highly coiled structure, and this is called proximal convoluted tubule. Then this proximal convoluted tubule leads to a loop like a hairpin having the descending limb and the ascending limb. This is called the Henley's loop. Then again it opens into a coil structure at the distal end. So, it is called the distal convoluted tubule, and finally it opens into a collecting duct which receives many nephrons. So, this is the structure of the renal tubule. Now, the second structure is the glomerulus. The glomerulus is a tuft of capillaries, a cluster of capillaries in the concavity of the Bowman's capsules. In the cup or in the concavity of the Bowman's capsule is a tuft of capillaries called the glomerulus. The blood vessel entering the glomerulus is called the efferent arteriole, and the blood vessel emerging out is called the efferent arteriole. Don't forget, efferent is the blood vessel entering the glomerulus and breaks up into fine capillaries which forms a network or a cluster in the Bowman's capsule, and this is called, this cluster or tuft of blood capillaries is called glomerulus. And the blood vessel that emerges out is called the efferent arteriole. Now, here there is another structure to be identified. This in totality is known as the Malpigin tubule. The Malpigin body or the renal corpuscle. So, what is renal corpuscle or Malpigin body? It is the Bowman's capsule along with the glomerulus forms the Malpigin body or the renal corpuscle. Now, continuing with the efferent and efferent arteriole. So, I told you the blood vessel that enters is called the efferent. It breaks into a cluster of capillaries called glomerulus, and the blood vessel that emerges out is called the efferent arteriole. It is not over. This efferent arteriole that comes out breaks into a network of capillaries that surrounds the entire nephron, that wraps the entire nephron. So, it is going to break up into a network that surrounds the entire nephron. So, I am wrapping it. I am not scribbling, I am just wrapping it. And you can see one branch of this blood vessel runs parallel to the loop of Henley, and it is also U-shaped. Like the loop of Henley, it is U-shaped and runs parallel to the loop of Henley or Henley's loop. See, I am highlighting it. That is a branch of this blood vessel. This blood vessel is called, loop shaped blood vessel is called vasa rectae. What's a blood vessel called? Vasa rectae. And this entire network of blood capillaries wrapping the nephron is known as, never forget, PTC, Peritubular Capillary. So I told you the efferent arterial which emerges out breaks into a number of capillaries and wraps the nephron. That blood vessel is known as the peritubular capillary. From the meaning you will understand, it's on the periphery of the renal tubule, and that's a capillary, peritubular capillary. And one branch of this peritubular capillary which runs parallel, U-shaped, and runs parallel to the loop of Henley is called the vasa rectae. Okay. So with this, the structure of nephron is complete once again. So once again, reiterating the structure of the nephron, there are millions of nephron in each kidney, and the structure is basically divided into two parts, one glomerulus and two the renal tubule. As I told you, the renal tubule starts with the Bowman's capsule and ends at the collecting duct. So I am going to draw only the renal tubule. It has a double wall cup called the Bowman's capsule, which goes to a highly convoluted structure called the PCT, which leads into a hairpin loop called the Henley's loop, having the descending limb and the ascending limb, and then another convoluted tubule called the DCT, which opens into a collecting duct. This collecting duct receives many such nephrons. Okay, so this parts I am indicating here: Bowman's capsule, PCT, Henley's loop, DCT, and collecting duct. These are the parts indicated of the renal tubule in green. Now the glomerulus. As I told you, glomerulus is a tuft of capillaries in the concavity of the Bowman's capsule. The blood vessel entering is known as the efferent arterial, and the blood vessel leaving or emerging is called the efferent arterial. This efferent arterial breaks up into a network of capillaries that wraps the nephron. This blood capillary is known as peritubular capillary, and one branch of this peritubular capillary is running parallelly to the loop of Henley, and it is also U-shaped, and that is called vasa rectae. Also I told you about one structure. What is Malpigin Corpuscle? So Malpigin Corpuscle is this region. It is Bowman's capsule plus the glomerulus, which is found in its concavity, is called the Malpigin body or the renal corpuscle. There are two types of nephrons, namely cortical nephron and medullary nephron. The actual name is juncta medullary nephron. Let's find out their difference. So to know their difference, I am going to redraw the kidney, a rough diagram of the kidney. This is a kidney. The violet is indicating the cortex. It's almost done, and let yellow indicate the medulla. Okay, so we have seen two zones or regions in the kidney, namely the cortex and the inner medulla. Now I am going to place two sets of nephrons, symbolic representation of two nephrons in white color, so it stands out. So the nephron what I am going to depict is like this way, like this, then the loop of Henley, and like this. So these I am going to draw two sets in white color, so it stands out. See this one like this. I am going to draw in green color because white, the board is not taking white. So in green color I am drawing some nephrons. Now what is indicative here is a Bowman's capsule. It's becoming another color. Okay. I hope you can see this. This is the Bowman's capsule. Then this is the PCT, DCT, and so I drew one nephron. The second nephron I drawn adjacent. Now you can see the difference. Can you see the difference between the two nephrons? You can see if this is nephron A and this is nephron B, you can see nephron A has a very long Henley's loop. See, very long Henley's loop that is dipping deep into the medulla. Whereas in nephron B, you can see the Henley's loop is very short. It's too short. It does not, just dips into the medulla. It's very short. So one difference here I have highlighted is with respect to the length of the loop of Henley. In A, the loop of Henley is very long, going very deep into the medulla. I am exaggerating, going so deep into the, dipping down deep into the medulla. And the other set of nephron, it has very short loop of Henley. So now which will be called the medullary nephron and which will be called the cortical nephron? The B will be called cortical nephron and A will be called the medullary nephron. So first difference between medullary nephron and cortical nephron is with respect to the length of the Henley's loop. In medullary nephron, the loop of Henley is very long that it goes deep down into the medulla. On the other hand, the cortical nephron has a loop of Henley which is very short. The second difference is with respect to the blood vessel. That blood vessel for medullary nephron, the vasa rectae is present. The blood vessel, I told you U-shaped blood vessel which is parallel to the Henley's loop is called the vasa rectae. The vasa rectae is present for the medullary nephrons which is running parallel to the Henley's loop, whereas here it is, if it is present, it is highly reduced. Okay, it is absent or reduced in the cortical nephron. So second point is with respect to the vasa rectae. Very prominent vasa rectae in the medullary nephrons, whereas in the cortical nephrons they are highly reduced or absent. Last, can you guess which was more in our kidney? Is it the cortical nephrons or the medullary nephrons? Which type is more? It is the maximum number is the cortical nephron. See, I put the symbol. Which is more in number? Majority of our nephron is the cortical nephron. Okay, so this is maximum. The cortical nephrons are more in number than the medullary nephron. So we have more of cortical nephrons than the medullary nephrons. So once again, the three differences between cortical nephrons and medullary nephrons. If I am going to write it here. First difference is with respect to the Henley's loop. Second difference is with respect to the peritubular capillary called vasa rectae. And third is with respect to the numbers. So let's start. Just three differences. Okay. So I have the cortical nephron here and this is the juxtamedullary nephron. Okay, these two are the two types of nephron. The first difference, Henley's loop is very long for juxtamedullary nephron, it's very short in cortical nephron. So Henley's loop. Then vasa rectae, it's reduced or even absent in the cortical nephron, whereas here it is very prominent. It is present for the juxtamedullary nephron. And lastly, the numbers. This is enough. There's more cortical nephrons. Cortical nephrons are more than the juxtamedullary nephrons. So this was why I was trying to bring out the kidney to explain the location and to get the concept clear. So just remember, there are two types of nephrons, cortical nephrons and juxtamedullary nephrons, and the three difference with respect to the Henley's loop, vasa rectae and the numbers. So I hope today's slot regarding the classification of animals based on nitrogenous waste, the human excretory system, the structure of kidney, and finally I ended up with the structure of a nephron is clear. Okay, it's very, very important, and this is actually depicting the actual structure. The structure which I drew is an exaggerated version to show all the parts clearly. Okay, I hope this video is clear to you. Thank you.